So hello to everyone. First, I would like to thank the steering committee for inviting me to present our data on the links between metabolisms and tumorigenesis in the liver of glycogen storage disease type 3. This work was performed in collaboration with the team of Giuseppe Ronzetti at Geneton in Paris and my lab in Lyon, named Nutrition, Diabetes, and the Brain. So first, have a look to the clinical picture of GSD3 patient. So the first clinical manifestation appear in the first months of life with hepatomegaly due to the accumulation of glycogen in the liver and short fasting hypoglycemia, but without hyperlactatemia, contrary to what is observed in a patient with GSD1. The patient with GSD3 also accumulate glycogen in muscle that leads to muscle damages, which may manifest itself as abnormal fatigability from childhood that evolve in some cases in muscles weakness. With time, the impact of which on daily life is very variable, but may go as far as disabling myopathy with impossible autonomous walking. In addition to muscle weakness, cardiac damages, which are mainly hypertrophic cardiomyopathy, may be present early even early in childhood. Finally, with time, the liver pathology can progress to cirrhosis and even in hepatic tumor. At the histological level, we can observe at an early stage the development of fibrosis, as you can see here, in the liver of a young patient with GSD3 after a trichome staining that reveals the accumulation of collagen fibers in the liver. It is a link to an increase in plasmatic transaminases due to ongoing hepatocellular damages. Some data in the literature show that cirrhosis can be detected in older patients, and a recent report indicates that nearly half of patients with GSD3 over 40 years old develop hepatic cirrhosis that can progress to decompensated cirrhosis requiring liver transplantation. As a patient live longer and longer thanks to their nutritional management, the development of hepatocellular adenomas and carcinomas is observed more and more often. And this recent publication shows that hepatocellular carcinoma occurs in more than 10% of patients at a median age of 60 years old. So to better understand the molecular basis of the development of this tumor, we have developed a mouse model that reproduces the pathology. So this mice have a constitutive deletion of AGL exon 6 to 10 and were backcrossed onto a mixed B6 bulbous C background by the team of Giuseppe Ronzetti, and the phenotype of young AGLKO mice, so at the age of 3 months, was described in 2018. First, we showed that the loss of AGL gene was total in the liver and the muscle, and this led to the accumulation of glycogen in the liver that that is responsible for an increase in the liver weight in KO mice compared to wild-type mice. In addition, glycogen is also accumulated in the muscles, including the heart, and this leads to a muscle weakness even at the age of 3 months. In addition, AGO mice show fasting hypoglycemia, and we also observe decrease in blood glucose at the phase state in AGLKO mice compared to wild-type mice. So here we are focusing on the liver pathology, and our main question is what is the impact of the loss of glycogen debranching enzyme on the liver metabolism, and especially at an advanced ages. So we studied the liver metabolism in HGL-KO mice at the age of 14, 15 months. So first, we confirmed the accumulation of glycogen and hepatomegaly in this set of mice, of cow mice, in response to the blockage of the glycogen debranching enzyme activity. Here, we must highlight that that glycogen accumulated in GSDIII liver has a specific abnormal structure characterized by short chains, and this glycogen is called limited string. So the blockage of glycogenolysis led to a dramatic drop in intracellular levels of glucose 6-phosphate and glucose, and this leads to a decrease in glucose production by the liver. Indeed, HGL-KO mice show a lower glycemia compared to wild-type mice, and however, we can notice that hypoglycemia was less severe than what we generally observe in mice with GSD1A. Indeed, glucose production, blood glucose levels was not lower than 50 milligram per deciliter, thanks to significant glucose 6-phosphatase activity. As you can see here, glucose 6-phosphatase is lower than wild-type mice, but allows the mice to produce glucose. So then we observe that we observe a decrease in glycolysis in GSD3 liver. Indeed, the expression of the main enzyme of glycolysis, such as glycokinase and pyruvate kinase 1, was decreased compared to wild-type mice, and this led to a decrease in pyruvate oxidation and a decrease in the lactate production by the liver. Finally, in accordance with the decrease in glucose 6-phosphate level that lead to the inhibition of glycolysis and pyruvate oxidation, we also observe very low levels of hepatic triglyceride content and cholesterol content. And this was confirmed by an inhibition of the expression of enzyme involved in de novo lipogenesis. So all this data suggests a low energy status in the liver of GSD3 mice, characterized by low glucose, low lactate, and low lipid levels. So despite this low metabolic condition in energy substrates, GSD3 mice developed hepatic tumors at around 15 months of HGL deletion, and this was correlated with an increase in alpha-fetoprotein plasmatic levels, which is biomarkers of liver cancer. On average, 30 to 35% of mice develop hepatic nodules, which were generally characterized as hepatocellular carcinomas. And in an interesting but unexplained way, hepatic tumors were more frequent in female than in male. So as expected, the tumors were developed on hepatic fibrosis. As you can see here after red cyrus staining. And in parallel, we also observe an increase in the expression of pro-fibrotic genes, such as TGF-beta1, PI1, and collagen-1, and an increase in plasmatic transaminases. So to summarize this data, let me compare the liver metabolisms and tumorigenesis in GSD3 versus GSDIII. So first, in both diseases, glycogen is accumulated in the hepatocyte. However, in the case of GSD3, glycogen is abnormally structured that can induce cellular stress in the hepatocyte. Glycogen accumulation rapidly results into hepatomegaly in GSD3 and GSD1, and this is a characteristic of all hepatic glycogen storage diseases. Then our data, our results have highlighted two major differences. First, GSD1A are steatotic liver, while no lipids are accumulated in the hepatocyte of GSD3 mice. And second, GSD3 livers are fibrotic, while no low grade of fibrosis is observed in GSD1 livers. Finally, these two metabolic different conditions lead to the long-term development of tumors, but we can note that the development of hepatic tumors is more frequent in the metabolic context of GSD1A than in GSD3, since we can observe about 70 to 80% of GSD1 patient who develop hepatic tumor, while only 10 to 15% of patients with GSD3 will develop hepatic tumor at an advanced age. Thus, molecular mechanisms of tumorigenesis should be quite different in these two pathologies. So to go deeper, we recently show that in GSD1A, the absence of glucose 6-phosphatase activity leads to an increase in glucose 6-phosphate level, and this metabolite is a central hub for the liver metabolism. Indeed, the increase in glucose 6-phosphate is responsible for the activation of glycogen synthesis, but also glycolysis and de novo lipogenesis, and this results in the production of biomembranes, carbon skeleton, and nucleotides required for biomass and DNA replication, and leading to a cell proliferation. So interestingly, this metabolic reprogramming that occurs in GSD1 liver is very similar to that observed in cancer cells and characterized by a Warburg effect. On the contrary, in GSDIII, the loss of the glycogen debranching enzyme activity leads to a dramatic drop in glucose 6-phosphate, favoring low energy status in the hepatocyte. So one important question is how cancer occurs in GSD3 hepatocyte, and what are the sources of energy to sustain cell proliferation. So in conclusion, we expect that comparing tumorigenesis in GSD1A and GSD3 can lead us to find specific therapeutic target to prevent the development of liver damages. Finally, I would like to thank all members of my lab and the team of Giuseppe, particularly Vajay and Luisa, who perform all the experiment. And many thanks to our funders, especially to the Patient Association that have supported our research work for many years. And thank you for your attention.